Good morning, everyone, and welcome to the Department of Laboratory Medicine and Pathology Grand Rounds. Today, it's my pleasure to introduce not one, but three speakers who will be talking to us about the use of artificial intelligence to classify kidney stone composition. Our first presenter will be Dr. Paul Janetto, who is an associate professor in DLMP and co-directs the clinical mass spectrometry, the clinical and forensic toxicology, and the metals laboratories. And he also serves as the DLMP Vice Chair of Supply Chain Management. Dr. Janetto is board certified by the American Board of Clinical Chemistry and the American Society for Clinical Pathology, and he's very active in his field with numerous publications and active service, including, for example, currently acting as the secretary on the board of directors for the American Association of Clinical Chemistry. Our second speaker will be Dr. Ricky Carter, who is a professor of biostatistics and serves as vice chair for the Department of Quantitative Health Sciences at Mayo Clinic in Jacksonville, Florida. He is also the director of the Biostatistics, Epidemiology, and Research Design Resources in Mayo's Center for Clinical and Translational Science, and is the scientific director for the Digital Innovation Lab, also on Mayo's Florida campus. Dr. Carter has also been very active academically and has keen interests in the development of digital biomarkers using machine learning techniques. And then our final speaker for today will be Mr. Patrick Day, who is a senior developer in the metals laboratory working with Dr. Janetto, and he holds the academic rank of instructor in the Mayo Clinic College of Medicine and Science. And while in the metals laboratory, Mr. Day has developed numerous LDTs and has managed a variety of multidisciplinary research projects, many of which have led to peer-reviewed publication. And he was a recent recipient of the ASCP 40 Under 40 Award. So congratulations for that. So I want to thank all three of you for taking time to speak with us or to us today. And Dr. Janetto, I believe you are starting first, so I will turn it over to you.
Thank you very much, Dr. Thiel. Good morning, everyone. It's my pleasure to be here as I'm joined with my two colleagues, Dr. Ricky Carter and Patrick Day. Today, as we talk to you about the classification of kidney stones using artificial intelligence. And Pat will share with you some of the data that we've actually collected over the past 12 months and really kind of implicate how AI can really help improve quality and patient safety. I have no formal disclosures. So over the course of today's talk, through the three of us, we're going to cover the following learning objectives, starting with discussing the clinical utility of kidney stone testing, how we do it and the workflow. We'll discuss the opportunities for computer vision techniques and machine learning and spectra classification. It will also describe the benefits of using artificial intelligence as a laboratory quality tool as it relates to the analysis of kidney stone composition. So I'm going to go first today and really just give you a background on kidney stone testing, the significance of it and how it's performed. Dr. Carter will then jump in and talk about the algorithm development and validation. And finally, Pat Day, who's really the brains behind this initial concept, is going to talk about the implementation of AI as a quality management and clinical application tool in the laboratory. So kidney stones are quite common worldwide. In fact, it's estimated that about 1 in 10 people in the United States will develop a kidney stone in their lifetime. And that includes up to 12% of men and 5% of women by the age of 70 will have one stone. Now, the problem is that those who do form stones have a very high reoccurrence rate of forming a second stone. In fact, it's about 50% of people will produce a second kidney stone in the next 5 to 10 years. And so with epidemiology showing an increase in the frequency of kidney stones, and if you've ever had a kidney stone, you'll know that these are very painful events. The analysis of kidney stones becomes a very important clinical test. In fact, after the stone passage or surgical removal, the accurate identification of the stone composition is probably one of the most crucial laboratory diagnostic procedures for both the treatment, but also that reoccurrence of that next stone. And so here at Mayo Clinic, we're proud to offer kidney stone analysis testing. We actually do over 90,000 kidney stones per year, and that number continues to increase. And as part of testing, obviously you have to have good samples coming in. That means proper collection, handling, and labeling of kidney stones. And so we do offer a kidney stone analysis kit for our patients and our clients. So if they do pass them at home, it comes with a kit with a filter, so you can catch it as it's passed. And then there's some procedures that have to be followed, because we do want that stone material to be clean of any blood or foreign material. So typically rinsing that with water is what we're looking at, and then drying that stone. So drying it at room temperature, not using any heat or elevated temperatures, as that can actually alter the stone composition as well. And then it would get sealed into the screw cap plastic container and indicate that source of that stone. Unfortunately, to date, over half of the stones that the laboratory receives look like the one on your left, wet, in urine and liquid. And unfortunately for us, as I'll show you later on during the analysis, that's problematic for us. And so we don't want to receive wet samples. They actually need to be clean and dry in order for us to get an accurate analysis of the constituents. So how do we analyze stones? Well, there are a number of modern methods, including things like infrared spectroscopy or X-ray diffraction. However, our laboratory actually uses what's considered the gold standard or reference method. We use Fourier transform infrared spectroscopy. And so the way this works is that once we get a stone in, that clean and dry stone, don't forget, we'll then crush it up manually. We'll add that to the FTIR instrument. An infrared beam then passes through the crystal of the powdered kidney stone sample. And during this interaction, the molecular bonds within the different constituents are going to absorb some of that radiation. And that's going to produce a very unique fingerprint or identification of the constituents. And so by looking at these different unique spectrum, this is how we can identify the different constituents in that kidney stone. And so here's an example of a typical calcium oxalate monohydrate stone. And you can see the various shapes and peaks. Remember how I said it has to be dry? If we have a wet stone, so even one that is not sitting in water but visually looks dry, when we crush it and when we put it, we'll see this huge absorbance peak here in the beginning of the spectrum. And that actually obscures and really blocks our ability to see the rest of the individual analysis and peak because of this strong absorbing peak. And so this is why we have to then go back and dry the stone ourselves and then do a reanalysis in order to get the accurate peaks so that we can actually identify the constituents in the stones. So one thing I hope you're starting to realize is this entire process is very manual. From the tracking of the kidney stones into our lab, accessioning them into the LIS system, manually crushing each one. Remember, these are irretrievable samples that can be very small to very large. And so we're manually taking these, crushing these, analyzing that powder using the Fourier transform infrared spectroscopy, manually looking at spectra. And we do have library spectra and things that we can overlay and help match, but it's still a visual interpretation. And then these results are entered into the LIS manually. And because this entire process is manual, we have a secondary technologist review that to make sure that what was put in matches the spectra before these results get released to the provider. So when we do this analysis, what are the types of stones that we do see? Well, the vast majority of our stones are calcium oxalate based. And then you can see the next most abundant comes into the hydroxyapatite, which is a form of calcium phosphate. We have uric acid stones, some struvite, and then lastly, some brushite and others that aren't listed on this slide. Now, what's the importance, though, of identifying all these individual components? Well, one of the things I told you is that we want to prevent that reoccurrence of a secondary stone. And so based on the constituents, we can understand the underlying cause or mechanism of why that stone formed, and also then the nephrologist can choose the appropriate treatment. So in the case of a uric acid stone, we would then want to alkalize a patient's urine to increase uric acid solubility. Or in the case of a hydroxyapatite stone, with an underlying cause being the hypercalciuria, how we could acidify that urine and therefore increase calcium solubility to, again, prevent that future stone formation. And again, calcium oxalate stones being one of the most common, obviously there's some other things we can do from a lifestyle perspective, is increasing daily fluid intake and also reducing calcium intake. And so there can be some lifestyle changes in addition to different treatments and or medications that could be prescribed. Other stone types that we commonly see are what's known as struvite or magnesium ammonium phosphate. The underlying cause of these type of stones means the patient has an infection. And so obviously here, the goal would be to treat the underlying urinary tract infection, and that would prevent this type of stone formation. It should be noted that a number of over-the-counter as well as prescription drugs are also known to sort of precipitate out or cause kidney stones. So shown here is ephedrine, guaifenesin. These are just some examples. We can actually see the spectra of these drugs. And so on our FTIR analysis, we can identify stones that are formed by various medications and drugs. And obviously from that standpoint, when we identify that, that would be a case where then you'd want to maybe alter therapy or change the patient's therapy so they're not taking that drug. So again, they don't have that predisposition to form a secondary stone. So in summary, I hope that I've quickly shown you is that we use the gold standard methodology here at Mayo Clinic for your transform infrared spectroscopy. They must be clean and dry for accurate determination of the kidney stone composition so that we can understand the underlying cause and appropriate treatment. And that while this is the most crucial laboratory diagnostic procedure for the treatment and recurrence prevention, it's also very manual. And so you can see where this might lead us to the development or use of something like artificial intelligence to help us. And so now I'm going to turn it over to my colleague, Dr. Ricky Carter, who will take the next part of the presentation.
So thank you. So I'm going to take you on a quick journey. Won't get into a lot of technical details. So I thought where we'd want to start is to train you. I think we all need to learn how to classify these kidney stone spectra. So I put together just a very brief quiz. So as mentioned before, this is the most common stone type. You've seen an example of this before. Take a moment and burn this into your memory and say, this is the spectra for a calcium oxalate monohydrate stone. The next most common stone we see is uric acid, 100%. And these 100% means that they're pure. They're not a blended stone. So if you look at this, the signature has some similarities, but there's some clear differences. You could look at these and differentiate it. As we move through the training, here's the calcium oxalate monohydrate again. Here's the uric acid stone, calcium oxalate, uric acid. So at this point, we're ready. And we're not going to use chat. We could do this. So if you can, mentally cast your vote. Here is a candidate spectra. It had been dried. It had been run through the FTIR scanner. You're looking at the display readout, and your job is to classify this with high accuracy. So I think many of you would look at this and you're counting peaks, you're looking at profiles, and you might say it's a calcium oxalate monohydrate stone. And what I would say is, sorry, you missed it. That there was 10% of this dihydrate compound in there as well. And so it's a blended stone. Now, in fairness, as we've worked with the clinicians, they would say, well, these would be clinically equivalent. You could allow 10% of dihydrate to be present and still be clinically in the same diagnosis. But what I'd like to show is that this problem of identifying some of these trace elements within the kidney stone spectra is quite challenging. So here's an example where brushite, instead of calcium oxalate monohydrate or dihydrate, is present. And what you see is that vast majority of the points in the spectra are identical, as you'd expect. But there's a couple of really, subtle differences in these stones. And you have to look closely to make this classification. So this is part motivation of the AI. How do you do this at scale? You need this double check that the practice uses. But I think as you start stepping back and looking at more generally, we've talked about how manual the process is. There's a drying component, there's a preparation, there's having a sufficient amount of sample to use on the scanner. And then there's simply natural variation. There's scanner drift, there's things that happen over the course of the day. And these are all calcium oxalate monohydrate stones, but you can see there's a fair bit of variation. Just from the laboratory practice, sometimes some of the stones are rerun. And what we've discovered in some of our algorithms is some of those previous, generated spectra get a correct label, but they're not good representations of the spectra. But this process of classifying the spectra is quite challenging. And so we're trying to develop tools to augment the human through this process. Now, the business needs clear that the number of stones continue to increase. We have 2 human bodies, and I don't know the job market and people that want to read kidney stone spectra, but across the world, there seems to be a labor shortage. This is a highly nuanced task, and thinking we're going to be able to just put more people onto the processing line may not always be tenable. We do know that there's a lot of very easy to recognize common stones. And if we had a tool that could maybe split the distribution of spectra into things that could be automatically classified and allow the humans to focus a little bit more on the more unique and rare stone types, that would give more value to the humans. So what I showed in that quiz, and I think the way that if you learned or if you were able to guess that it was a calcium oxalate monohydrate stone, you did a form of sort of machine learning in your mind. You did some pattern recognition. This is a diagram that's used in some fields. It's not without limitations, but it really goes to this talk to really help emphasize the point. When you think of a traditional statistical model, you have rules and data, and what you try to do is come up with some type of optimized solution. Rules in this case means the functional form we put on the data to make the math work out. So you might be counting peaks, you might look at locations of the peaks, and you just try to define those, and then you fit a model. The more generalized approach to this, and this is where machine learning comes in, is you don't really set any of these rules. What you do is you present the data and the answers, and you try to iterate over what you're seeing and come up with a set of rules that is the output that does the classification for you. So those shapes look different. You saw that there were differences. You might not be able to enumerate all the reasons why you thought they were different, but in your mind, you were quickly able to differentiate the 2 spectra. Now, I'm going to teach a little bit about convolutional neural networks. This is meant to be introductory, and I think in an example, what convolutional neural networks work on this mathematical principle called a filter. And I'll define that a little bit more mathematically in a bit, but you can think of this very simply as a board game. A game, Perfection, is a game I enjoyed as a child. If you're not familiar with this, it's a race against the clock. You push the board down, you set the timer, and you try to solve the puzzle by putting the pieces into the array where they belong. I enjoyed this game. It was fun. It wasn't really until recently I learned there was a second edition to Perfection, sort of a kiddie game, far simpler, bigger pieces, much more distinct sides. What I'm going to show is this is an example of how computer vision can be trained. So imagine a case. You put the 2 boards out, you put all the pieces into a bag, and you're completely blinded. You reach into the bag, grab a single piece out. Your job is to classify, is this the semi-adult version or the baby version of Perfection? And with a single piece, by determining if it matches in that array, you can correctly classify whether it's one of the 2 games. That's the idea behind some of these computer visions. You're looking for distinct patterns, checking to see if they exist within the image, or in this case, the game, and making predictions based on the accumulation of data. So this accumulation of data works in this way, and a convolutional neural network works through multiple processing steps. The first is represented by these blue things are filters. It's these unique patterns you expect to see. In this case, there's a dozen or so filters that have been defined, and the training part of the algorithm teaches the network what those filters should look like. You go through a series of calculations, and then at the end, it's just an activation that looks like a regular regression model. So SPECTRA comes in, these filters do the puzzle piece check, do some mathematical calculations, and makes a classification. So let's look a little bit deeper under the hood. So what you might look at is, so here's this tricky spectra that have this blend of 10% dihydrate calcium oxalate. You can look at some of these curved places and start saying, you know, one filter, one puzzle piece, if you will, might be the descending part around, I think it was about 100 nanometers. So what could you do? If that's your filter, you can go through and start representing this mathematically. And in this case, I'm going to let the blacker, bolder vision, sort of where the signal is, be a higher numeric value. And where this whiter the background is, let it be 0. A mathematical way to test whether or not two matrices are alike is to do what's known as a dot product. So you just basically multiply cells and add them up one after the other. If the shapes are similar, in this case, we have two different representative shapes, the dot product will become very large. So that's an impulse. So if something starts looking like it's matching, it generates an impulse. Those impulses are what's passed through the network and ultimately goes on to provide the classification. We also were challenged with a couple other aspects within the study. So unique to this study is we had a truth library. We had, you know, laboratory created spectra that were known in purity in the composition. Well, could we use our network to help identify in terms of a careful matching of spectra? So one way to think about it is this, you know, an observed spectra produces some type of digital fingerprint. We could run multiple spectra through this and generate a series of fingerprints. And then much like fingerprint matching, we can do some simple distance calculations and identify how close observed spectra are to a reference spectra. This was an insight. This was how do we leverage our institutional sort of IP around the reference spectra, but also provide a secondary check to the signal quality of the spectra. These things should, as is drawn here, be pretty coincident. So as we started building this model, you know, the idea is that we have an input of the spectra. That's the only thing we're using. We have algorithms. Our goal was to provide some type of auto pass triage, what's high, low accuracy. And so we had to develop different approaches. You know, there is an opportunity to think about in machine learning, you know, just let the computer learn everything it needs to know. Model 1 is about as close to that as possible. We had 708 unique stone profiles when we did the training. We've since discovered some more. And so some of this is we have to create algorithms that you know, stand the test of time. We decided to supplement Model 1, which actually had really robust classification accuracy, with some specific algorithms that met some of the target scope. You know, previously we talked about the individual constituents or stone composition having an indication on treatment. And, you know, one example would be, is there a presence of struvite in the sample? So we trained two different types of models to detect, you know, whether or not there was an individual constituent present. We had a binary. Do we think it's there or not? And then the secondary one, like what percentage do we think it is? And that's the shape of the overall spectrum. And then we included a fourth model, which allowed us to do some reference matching and spectral matching as well. So we put this together. So we've trained algorithms, we retrained algorithms, we evaluate results. We get to this point now where we're trying to fine tune what stones and what level of quality we want for the auto pass. There's a couple decision points here that are beyond, say, the machine learning. Is like, which stones do we actually want as candidates? You know, so calcium oxalate from the beginning was one we wanted to be able to pass. So this algorithm or this dashboard allowed us to tune the individual prediction parameters around each stone type to say, how certain do we want it to be and how close does it match the reference spectra? And if it's not really certain or it's not close, then it's rejected. And we can look at the overall performance here. And in this case, we were able to get 43% of all stones to pass through the computer network with an accuracy of 99.97%, which is quite robust. But we still have to step through this. We have to evaluate, and Pat Day will talk a little bit more about that in a moment. I want to show you a couple of examples of what the models are doing. So part of this is that there's a reputation for AI algorithms to be black boxes. Hopefully, as a part of this talk, we can talk a little bit of, shine a little light within the algorithms. They are computationally intense, but you can trace each step of the calculation through the network. We build dashboards to visualize the model outputs. And in this case, you can specify a barcode that's from the lab. It'll display the spectra. It'll display, some of the predictions of that model. And so in this case, the model is, giving 82% weight to a straight calcium oxalate monohydrate stone, which happens to be the correct classification. It shows there's some other potential to have some mixed composition, maybe that COM 90%, COD 10%, is about 17.9% of the spectra. The algorithm seen that looked like this also maybe had some dihydrate mixed in. We also did, as we mentioned, these stone-specific analysis or constituent-specific analysis. And you can see from this display, there's a little bit of a signal around dihydrate and a little bit of a more pronounced signal of the majority being the monohydrate. There isn't a lot of other signal anywhere else. And so you could probably safely conclude, it's a combination, maybe a majority of calcium oxalate monohydrate with a small composition of dihydrate. This is a more challenging spectrum. This is one where we got it wrong. But if you look at where it's wrong up there and under the classification, we just had the percentages wrong. The model got the right two constituents correctly, but it disagreed a little bit on the percent composition. You can dig in a little bit, and then you can see, what this model is also showing is that the flat distribution around the dihydrate, there's not a lot of certainty. My guess is there's a little bit of human preference in how these were coded within the laboratory. But interestingly, there's a small signal of appetite also present in the data. And if you look at the top 5 predictions, there is some mixing, but one of the top 4 or the fourth in the prediction does include a little bit of appetite. So this might give the case of this is a human review. And if you look at our first confidence, 0.216, this wouldn't meet our threshold. This would be something that the algorithms would say, we're not certain enough, send this to a human for review. Part of what we're able to do, some development from when we started, there's been a lot of work in computer vision and being able to overlay heat maps of where specific regions of an image or a spectra are triggering predictions. One of the more common tools now is this tool known as a SHAP explainer. What the two left and right columns working down is an input spectra. You can then see the pink bars represent areas that the algorithm is looking at in its effort to make a determination. So you see these peaks are what's showing up. That's good. It's not things off on the side. It doesn't look like it's things that are not relevant to the spectra. We could give this to the technologist, and this does match some of the peak identification that they're using to do their classification. And then what's interesting is this bottom photo or the bottom spectra is really another case of these blended spectra. And you can look and see how it takes on elements of both of those pure sources, and the activation and the sort of unique profiles is different. It's not exactly the addition of those two. The overall shape changes. And from a human perspective, it becomes a distinct shape that they need to memorize. We've been working diligently over the past year for sure, but we've been working longer than that with this idea of how do we start realizing the benefits from the algorithms. Our conclusion is it's easy to build a model. We built models that did some of the initial classification, in days, years ago. We were excited. We invested more and more time. But moving things into implementation is hard. And there's a little bit of a word soup there. It's a multidisciplinary activity. It requires engineering. It requires computer engineering around data flow, data processes. And to give a little bit of an idea of some of the tools and some of the complexities, here's just a quick laundry list of some of the things we're currently leveraging. And then visually, this is what it's looking like. We have to start mapping out the flow of information and building some robustness. And we've gotten better over time, but these things need to happen as any clinical system and be reliable. There's a lot of technology, there's a lot of security updates, there's a lot of moving parts. And so our current workflow has had breakages as things get updated, things change, permission changes, network segmentation changes. And so our future state, which we're getting very close, is to containerize all of this and make it much more of a contained element that's in a universe to itself, where the input is simply, the specters as the scanner generates, and then we can write out some simplified predictions to the laboratory information management system. So with that, I will hand it back to Pat.
All right. Thank you, Dr. Carter. So it is now my pleasure to describe how we implemented these AI models into our clinical practice as a quality assurance program, how we want to expand the use of this AI trained model in the future. So briefly, in this presentation, we'll discuss how we established AI trained models in our lab by describing the study design and study results of a prospective quality assurance study. And then briefly, we will touch on future plans for this set of AI trained algorithms and how it should improve the accuracy and the efficiency of our kidney stone composition assay. So before I go into study design, I want to give an abbreviated history of our algorithm development and validation for this project. To train the algorithm, we used over 70,000 clinical kidney stone spectra analyzed from our laboratory over the years. To validate the algorithm, we used a separate validation data set consisting of over 16,000 additional kidney stone spectra. After seeing promising performance, we then used the set of algorithms to prospectively conduct a quality assurance study. As we'll discuss later, this lasted a year and included over 81,000 additional kidney stone spectra. So let's discuss the framework of our quality assurance program. We've seen a version of this figure earlier in the presentation. It depicts our current laboratory workflow. Now, however, we have added a few green boxes to the figure to depict how we're currently using the AI trained models as a quality tool. For this study, we had our technologists report out stone results just like they've always have. They follow the workflow detailed in the blue boxes. However, in the background, we implemented our algorithms as a quality tool. As you can see in this first green box, once the spectra was generated by our FTIR instrument, we automatically sent an extra copy of the spectra to our AI models. Here, the model interpreted the spectra. The next green box depicts a data pull that we did every single day from our LIS. We pulled all the stone results that were reported by our technologists during the normal day of clinical operation. We then created a program that compared the AI interpretation and the technologist interpretation for each of those stones. If the two interpretations did not match, they would be added to a list of discrepant stone results that would be reviewed by our laboratory twice a week. So now I'd like to highlight a few of the capabilities of a custom application that the Digital Innovation Lab built us for this study. This application allows the laboratory to analyze data related to AI interpretations and to review the list of discordant stones. For example, the application tracks how well our AI models identify the 20 most common types of kidney stones. Additionally, it provides us this discrepant list of stone interpretations. An example of a list is shown here. The LIS stone label is the technologist's interpretation of a stone. The AI stone label is our model's interpretation of that stone. The model predicted probability is the value or how confident our AI model is in its interpretation. Now, if a technologist did incorrectly enter in an incorrect result into the LIS, we can now view it and change it in a timely manner so the physician has the correct stone composition to impact treatment. So now that we had our quality assurance program in place and our custom application was working, we were ready to conduct a prospective quality assurance study. This included a control period and a test period. The control period included 1 year prior to our quality assurance program. From this time period, we reviewed laboratory events associated with our kidney stone analysis. These events were extracted from our laboratory event management system. The test period was the 1 year period in which our algorithms were used to detect incorrect results in our laboratory information system. If an incorrect interpretation was detected in the LIS by our algorithms, a laboratory event would be entered into our laboratory event management system. This way we could compare the number of events prior to and post-implementation of our AI-based quality assurance program. So while we have statistics pertaining to a lot of different kidney stone types, I wanted to focus on the 4 most prevalent kidney stone types for this presentation. These types are calcium oxalate monohydrate, calcium oxalate dihydrate, calcium phosphate, also known as hydroxyapatite, and uric acid. I'd also like to highlight a few accuracy metrics we used for this study. Concordance rate is the exact match between our AI interpretation and our technologist interpretation. Concordance rate past QA is the number of correct classifications after application of a few quality assurance rules. Think of these as comparable matches. For example, if a technologist interprets a stone as containing 50% uric acid and 50% calcium phosphate, an exact match would be that our algorithm calls that same stone 50% uric acid, 50% calcium phosphate. However, a comparable match would be our algorithm calling that stone 60% uric acid and 40% calcium phosphate. While these interpretations are not exact matches, they are clinically comparable and will result in a treatment that's similar for the patient. The percent auto pass is a subcategory of results that we made that included stone interpretations where our algorithm had a very high predictive probability of being correct. As you can see in this subcategory, the concordance rate past QA in both the validation phase and our quality assurance phase were very high. Here are some additional diagnostic performance measures for the 4 most prevalent kidney stone types during the test period. These include accuracy, sensitivity, specificity, positive predictive value, and negative predictive value. As you can see, most performance measures exceed 95%, which indicate that our algorithms were performing quite well at correctly identifying these 4 stone constituents in kidney stones. Let's now look at laboratory event data associated with our study. In this table, you can see 3 different reasons for a revised report. The first reason for a revised report is an error that was detected in the testing lab. The second reason is that a client has called us and thinks our interpretation is incorrect. Finally, the third reason for a revised report is that an error was detected by our algorithms. As you can see in the control period, we had 10 overall errors. However, when we used our algorithms to find errors in the test period, the revised report rate jumped to 78, with 62 of these being detected by our AI quality assurance program. This resulted in a nearly 8 times increase in the relative revision rate, with most of this increase being attributed to our AI quality assurance program. So now I'd really like to highlight some of the conclusions from the study. First, an AI-trained quality assurance program that reviews human SPECTRA interpretations can result in a significant increase in the detection of incorrect results in the LIS. Secondly, by finding these errors in a timely fashion, we were able to reduce the number of incorrect stone composition results that end up in the patient medical record. This will ultimately improve patient care and safety since the physician can now treat the patient according to the correct stone composition values, which are so important for the effective treatment and prevention of kidney stones. Furthermore, this has the potential to reduce healthcare costs associated with erroneous laboratory results. So with the last couple of slides, I would just like to discuss at a high level our next plans. As you recall, we have seen this figure before. You can see that the vast majority of kidney stones contain calcium oxalate, hydroxyapatite, also known as calcium phosphate, and uric acid. As our quality assurance program has shown, our algorithms are performing very well at identifying these kidney stone constituents. So in the next phase of our project, we would like to push the algorithm to be directly integrated into our clinical workflow, instead of using it as a retrospective check of our technologists. Here's another figure that depicts our ultimate state. Instead of double checking the human interpretation that already appears in the LIS, we want our algorithms to identify most of our kidney stones right after it's generated by the FTIR instrument. In this way, we hope to improve the interpretations the first time improve our staff workflows, and potentially improve the efficiency of our workflows, since the models now will be making the primary interpretation for the majority of our stones, and our technologists do not have to manually enter in their interpretations into the LIS. Now, in order to make this future a reality, we understand a lot of things need to happen. For example, in order for us to successfully deploy AI-trained algorithms directly into our clinical workflows, we need more than just innovative algorithms. Just as important, we need innovative architecture or workflows. This new way of analyzing and reporting kidney stones will require things such as algorithm control and monitoring, scalable and secure cloud environments, flexible lab middleware, effective change management, and laboratory buy-in. As you can imagine, this is going to have to be a very collaborative endeavor that involves dedicated work and expertise from multiple groups, including the Digital Innovation Lab, DLMPIT, DLMPCPAI, Regulatory Affairs, and the Metals Lab. Thankfully, we work at an institution that is very collaborative and can make this a reality. So in conclusion, by using a suite of AI-trained algorithms, we have created a quality assurance program that can detect incorrect stone results in our LIS. We have created a user-friendly app that clinical laboratories can use to conduct quality assurance using these algorithms. But most importantly, we have shown that these algorithms are decreasing the number of incorrect results that ultimately end up in our electronic medical record, which should improve patient safety and quality. Finally, we're not stopping there. We hope to actively incorporate these algorithms directly into our clinical workflows in order to improve accuracy, improve staff workflows, and improve laboratory efficiency. Finally, I would just like to acknowledge a few of our great colleagues that have made this program a success. From the Metals Lab, Dr. Bornhorst, Sarah Erdahl, Denise Rocky, and Laura Schneider. From DLMPIT, Tim Alexander, Dale Parker, and Daryl Aubert. And from the Digital Innovation Lab, Miko Werzerek and Patrick Johnson. So now, with any remaining time, Dr. Janetto, Dr. Carter, and myself would be happy to answer any questions. Thank you.
Excellent. Thank you all very much. So we got a couple of questions in our Slido. And our first question here is, does the algorithm catch any pre-analytical issues that cause strange spectra?
So I think that could come to me. So yes, but we're always working to make that stronger. So we do have an inventory of what we call like bad spectra. And so part of what we want to do is train like a bad spectra identifier. We do simple checks on the range and making sure we get all the observed values we expect. There are some inversion, there are some strange things. So we do try to check that, but there's always more work to be done.
Excellent. The next question we have from the audience is, what acceptance occurred prior to allowing the human result to be revised? Sorry, this just got moved around. What acceptance occurred prior to allowing the human result to be revised to the AI result, or was the AI approved before starting to revise?
So I think I'll jump in here. But when in the quality assurance program management, we always, when Pat ran that discrepant report, we did have both Pat and Denise, I'll call out some of our expert manual review, go back, re-look at the spectra and make sure what the correct call was. So it did undergo a third review, so to speak, to determine sort of the tiebreaker, which one is correct. Was AI right, or was the original tech call the correct call?
Next question is, how long does the AI take to give a result?
So I think, to be honest, that's something we'll have to monitor going forward. So our workflow right now is a little bit asynchronous. You know, we're post-processing files on disk. We are building an API that's essentially going to make it real time. We just need to better understand what real time is. There's still some file transactions that need to happen, but it's measured in tenths of a second. So it's good.
I see. So nothing that is impacting workflow in a meaningful way.
No.
Excellent. Next question we have is, are there other institutions using AI for stones, or is Mayo leading this?
So I can take this. So in general, in the world, there's actually a limited number of laboratories doing kidney stone analysis. That's probably in the order of around less than a dozen just in the United States. And most of those places, to our knowledge, we are the first ones using AI to help and aid in the interpretation of kidney stones.
There
Excellent.
have been, I think, a few small case reports where people have trained some basic algorithms for small scale classification, but not to the extent we've done.
It's an interesting question that's been shared now is has the AI found anything brand new in terms of stones?
So
Yeah.
I don't believe so. I think it's an interesting use case in that,
Mhm.
you know, could you eventually get to a point where your algorithms would flag unique stones that they haven't, you know, been previously trained on? And could you then, you know, further study those stones and determine is there a new cause or is there something like that? But at this point now, we're kind of focusing on the, you know, those majority heavy hitters that will really improve our accuracy in our workflow.
And a question that I have is, how do you guys address education in this? Like, you know, we have residents and fellows training up. How do we train our learners to become effective users of AI or interface with AI effectively?
I mean, there are multiple attempts. So there is a master's in AI. It's geared more towards clinicians. There's board certification in informatics. It's trying to teach people to be a little more computational in the practice of medicine. I think probably the best way is to work hand in hand with the data scientists and you know, ask these questions of what is the algorithm seeing? Help me understand. And it's not that you have to trust the models, but I think understanding the models comes through these dialogues and changing some of the vocabulary used and understanding sort of the role of machine learning.
Yeah, certainly as we go forward in this pathway, there's a lot of conversation about AI in clinical practice, and it's going to be important to kind of integrate this with our training programs. Next question we have is the low number of labs doing stone analysis due to urinalysis crystal identification?
So there are a number of other tests that could be performed. So in the renal laboratory, even here at Mayo Clinic, they do the supersaturation test that can characterize the constituents of a urine, looking at things like oxalate concentration, uric acid concentrations, and things like that. Because let's face it, not all kidney stones are captured. Some are passed and flushed into the waste system. And so we don't always have stones to do analysis. And so there are other laboratory tests that can be utilized to understand the cause or underlying cause and therefore direct treatment strategies for testing outside of stone analysis.
Question that came in now is AI is often viewed as a replacement. Can you describe this use as more of an assistance to frontline bench staff?
Yeah, I mean, this is one of the major reasons why we came up with this idea. I know, you know, personally running kidney stones for years, it is very monotonous, it is very labor intensive, and it's something that, you know, one of the primary movers for this was how do we make it easier on our staff because it is manual and it's complex. So I think, you know, just showing them real life examples of, you know, we are human, we do make like interpretation errors, manual entry errors, and this is just a check and to make sure that the patient is getting the correct result. I think, you know, we've multiple times shared with our staff, you know, this is why we're doing this. It's not to replace you. And we really want to have you focus your time on these complex stones, some of these drug metabolite stones, or maybe some of these, you know, struvite stones where you need to spend a little more time on them. Let's focus there and, you know, remove some of the burden of some of these easier stones from your plate.
Yeah, I'll just echo that. I think it's very important to get staff buy-in. You know, in some areas of laboratory medicine, we already use things like auto verification, core lab, chemistry results and things are very commonly auto verified with rules. But in these newer applications in different areas, it really takes an understanding that we're not out to replace you. Computers are not out to replace us. It's not going to be a bunch of robots in the lab, but we want to do things more efficiently. And I think Pat hit the nail on the head in that we want to focus on those more challenging and difficult stones where you do need those little human nuances and capabilities. And some of these common ones that we can do automatically take some of that burden off of our staff, because I think we all know that currently there is a labor shortage. And so anytime we can help alleviate that stress on lab staff and use tools to help us produce accurate results, I think that's a great role for AI in the laboratory.
Excellent. Well, I think maybe if I can ask you guys to do a round robin and maybe just taking a step back and looking at this process and what you've done, I'm curious to kind of each of you to kind of share what's been your biggest surprise about this process and doing this. I don't know, Dr. Janetto, you want to go first?
Sure. So I think one of the things going into this, I will admit I am not an expert in AI, and it was alphabet soup. Dr. Carter was an excellent teacher. I had to learn a new lingo, right? Even in the laboratory, compared to clinicians, we sometimes speak different languages. And so understanding it. But then I think the biggest learning lesson, and this has been a multi-year progress, as we see the finish line now heading towards ultimate clinical implementation, but really the amount of collaboration and work between different groups to make something like this happen. And I'm very fortunate to be here at Mayo Clinic where we have all of these different expertise, like Dr. Carter and his team, like our Google engineers, like our LaPia, Sapia, all of these different groups who have played a role in ultimately bringing this forward, because it's really a team effort to get this to work. It's not like you can just plug and play, buy something off the shelf and it's going to work, but really implementing all of those different pieces. That has been an incredible piece for me as part of this journey.
It's awesome to hear that classic Mayo Clinic story. Dr. Carter?
Yeah, I don't know exactly if this fits the intent of your question, but I think that the thing that this study, attenuate in my mind is this idea that it's team science, multidisciplinary, but all that's built on communication and compromise. We each approached this with our own lens. We saw the solutions to this with a more narrow focus. And so this larger team and open discussion really helped round that out. So.
Excellent. Well, thank you. I'm going to have to hit you up for our Lab Medicine Rounds podcast to talk about that. All right, Dr. Day?
So, yeah, I would just, echo, Dr. Carter and even our colleagues in LPIA, years ago when we pitched this idea, it was kind of a slightly crazy fringe idea and we didn't really have any funding and we didn't really have much support, but Dr. Carter and his group hung with us for years of that, and same with LPIA, and that, they were willing to, spend their individual time, even though, we weren't, something flashy, kidney stones isn't the flashiest topic, but, they were willing to stick with us. And I think, we have a very cool product here that should, improve the accuracy of our results and ultimately improve patient care. So I think that's pretty cool.
That's awesome. It's great reflections from each of you. We had one more question come in. Is there a different cost structure or CPT code for the AI results? Does that change any of that at all?
No, at this point in time, no. There is no additional CPT coding or added cost or reimbursement.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.